Hello and welcome to Back to the Science. I'm Dr. Susan Oliver and I'm a scientist, and back there is my helpful assistant, Julie Oliver, and she's a doctor. In this video, we're going to be looking at some claims that were made on Dr. John Campbell's channel suggesting that Omicron was engineered using laboratory mice and then accidentally or deliberately released into the human population. Now, just to be clear, John didn't make these claims directly. He was reading out comments or emails that he had received from his listeners. But based on the comment section of his videos, it appears that a lot of people think this idea is viable. So in this video, we will be looking at the reasons why it isn't. But first, let's go back to the science and look at a previous video where John is explaining this paper here, which provides some evidence suggesting that the Omicron variant may have evolved in mice. Now, if you've seen any of John's videos, you'd know that he does a great job of making scientific or medical information easy to understand for people with non-science backgrounds. And this video was no exception. However, he did make a few mistakes which are relevant to the later claims that are made in the following video. So let's have a quick look at what he said and what he got right and what he got wrong.
We see that there's various mutants here, alpha, beta, gamma, that arose from this original strain here. This original strain is the B111, the original Wuhan strain. Then that evolved into delta, but we see that Omicron's on a completely different lineage. So Omicron did not evolve from delta. So where did it come from? How come we have it? And why is there such a long line there with a mystery unknown past of Omicron?
Now, John has pretty much got this right, except that the B11 variant is not the original Wuhan strain. The first B11 variant to become famous was what is now known as the Alpha variant, but was formerly known as the UK variant or the B117 variant. Both Omicron and Alpha came from the B11 variant, but other variants like Delta, for instance, didn't. The original Wuhan strain is just known as wild type or similar. And the strain that first spread around the world is known as D614G. And I should have said the variant that first spread around the world.
Well, the first hypothesis is that Omicron could have cryptically spread and circulated in a population with low levels of testing. So, for example, did it originate in an African population, for example, where people were simply not tested? So it grew and evolved in people, but it was never discovered because the people weren't tested. It's possible, but that doesn't explain why the mutation rate was particularly high. So that's the first hypothesis there. Could have happened, but doesn't seem likely because it doesn't explain the fast mutation rate.
This is a fair point, but it is worth mentioning that the overall mutation rate for Omicron was no higher than in other variants. It was just the spike protein where there was a faster mutation rate. Because SARS-CoV-2 is a novel virus, we really don't know enough to draw definitive conclusions.
Second hypothesis, Omicron could have evolved in a chronically infected person with COVID-19. Now, this is what we'd thought. So if someone is chronically infected with COVID-19, that they've got a compromised immune system and the virus will keep evolving to evade the limited amount of immune response that person is able to generate. And that is a reasonable idea. So second hypothesis, that it evolved in a chronically infected person and it arose in Africa. So, for example, did it arise in someone with HIV who's got chronic immunosuppression? That is possible.
I agree, this is definitely a possibility. And since John recorded this video, this paper has been published that provides some evidence that supports this hypothesis. They found that a number of the mutations that are found in Omicron also occurred in a variant from a patient with HIV who had a persistent SARS-CoV-2 infection.
But the third hypothesis is Omicron could have accumulated mutations in a non-human host and then jumped back to humans. In other words, it could be from animals. So this original Wuhan strain could have got into an animal group, or this mutation here could have occurred as time went on in an animal group, and of course we don't keep surveillance of all the animals in the world, so we wouldn't possibly have known that. It could have mutated away quite happily under the human radar and then just reappeared. And this is what I think is most likely. And this leads us on to this paper here. Now, peer-reviewed paper, Journal of Genetics and Genomics, and it's all there. Now, to be quite honest, this is not an easy read.
As we've previously discussed, it didn't come from the original Wuhan strain, but I do agree with John that the paper is not an easy read.
The likely candidate is mice. Now, the other candidates that were similar were camels, because we do know that Middle East respiratory syndrome comes from camels, and the other animals that were possible were actually goats. But mice were way out in ahead as the most likely contender.
Okay, so John has slightly misunderstood this. Camels and goats weren't likely contenders. The authors did some computer modelling after they determined that mice were the most likely host, as a check to see if any other mammals had similar predicted binding for their ACE2. They found that none did, but goats and camels had a similar predicted binding to humans. This figure shows the mammals that they did consider. They looked at mice, obviously, cats, dogs, cattle, pigs, bats, and camels. No goats. And this figure is the results of a data analysis technique known as principal components analysis. And they performed this on the molecular spectra of various coronaviruses that had been previously isolated from different mammals. Now, principal components analysis is used to identify what components of data are responsible for the most variance in the data. And after you've done that, you can plot your data based on the two main components, which is what they've done in this figure. And if you look at the figure, you will see a red dot that is labelled pre-outbreak Omicron mutations. And the red dot is kind of hanging out with the black dots that are associated with mice. They use more scientific terms to describe it, but that's what's happening. And that's what they've based their initial hypothesis that Omicron came from mice on. They have then done further analysis to bolster the evidence that it did come from mice. And we'll go back to John to talk about this bit.
The mutations that are there on Omicron are the mutations which would make the binding into the mouse ACE2 receptor, which of course is how the virus gets into the cell. The spike protein mutations on Omicron fit very, very well, almost perfectly.
John explains this really well. Omicron binds better to mouse ACE2 than other variants. And although the paper we are discussing only showed this with computer models, another research group has shown this experimentally. And this is the paper that showed it. But here's the really weird thing. Although it binds to mouse ACE2 better than other variants, it is less effective at actually infecting mice. And studies in mice like this one show that it leads to lower viral loads in both upper and lower airways compared with other variants. This is possibly related to the different cell entry mechanism compared with the previous variants. But that's just me speculating. I'm sure there's a bunch of scientists performing experiments as we speak to get to the bottom of it. And if you want to know more about the different cell entry mechanism, I covered in my video called Is Omicron the New Ivermectin? The question is, though, if the mutations in Omicron are a result of selective pressure from a mouse host, why is Omicron no good at infecting mice? There's possibly a good answer to this question, and someone is about to publish a paper explaining it, but at the moment, the mouse theory has lost a bit of shine.
It evolved to increasing binding to mouse ACE2 receptors. Of course, that's what the evolutionary pressures would exactly suggest. That's why it's consistent with human immune escape, because the human response is designed against the wild type for the vaccines, particularly the Delta type for people that have been exposed. But it's consistent with the human escape, and it's consistent with the upper airway adsorption of the virus.
Okay, so this isn't what the paper says. They actually say that the mutations associated with immune escape are additional to the mutations that are associated with improved binding to mouse ACE2. So adaptation to mouse ACE2 is not what is driving immune escape. Regarding being more likely to infect the upper airways, the paper makes no mention of this whatsoever. I can't think of any reason why binding well to mouse ACE2 would make Omicron more likely to infect human upper airways. But maybe John has some information from somewhere else that supports his statement. In the unlikely event that he ever sees this video, he could let me know.
Suppose this had gone into another animal that caused mutations to fit into that animal's ACE2 receptor, that meant that the Omicron variant was more likely to infect human lung tissue than infect human upper airway tissue. Then this Omicron variant would have jumped back into humans, but would have been more likely to cause more severe pneumonia rather than the upper airway infection it's currently causing. Then we could have been in trouble. So aren't we fortunate that it went into mice? That the nature of the receptor in the mice means that the virus, the Omicron, would escape the human immune system, that it would spread through all out humans, generating large amounts of immunity, but yet does not cause significant pathology in humans. And that is purely good luck, that it's not causing a 5%, a 10%, a 20% case fatality rate in humans, because Middle East respiratory syndrome does. It kills about 40% of the people that it infects.
Whilst I agree that we are lucky that Omicron isn't as deadly as MERS, we would be much luckier if it didn't exist at all. It has still killed a lot of people and hospitalised even more.
There is a slight piece of extra information I want to give you here. The science here was done at the Chinese Academy of Sciences. But to be fair, it did not say anything about the original viral origins, nothing at all. It's entirely about the origin of Omicron, and it has been peer-reviewed.
Now, I'm not sure why John thought this was relevant to mention this, but it certainly seems to have stirred up a lot of anti-scientist conspiracy theories from his viewers. And that's what we'll look at now.
Nicholas, talking about yesterday's video. As someone who has studied genetics and evolutionary biology, SARS coronavirus 2 is not behaving like other viruses in its genus, actually meaning the origins are likely from labs. Nicholas thinks this, I don't know. We need to consider this hypothesis very seriously because this is a potential for modern warfare and bioterrorism. It is really strange the way that Omicron has come to our rescue just at the right time.
Okay, nice appeal to authority from Nicholas. But John spent a whole video explaining in detail how the evidence was consistent with it evolving naturally in a mouse host. And he found the evidence pretty convincing at the time. And as I mentioned, there is also evidence consistent with it evolving in an immune-compromised person. If Nicholas thinks it was developed in a lab, he should point out the mutations that are consistent with this, not just make unsubstantiated statements. Regarding John's claim that Omicron came to our rescue at just the right time, I'm not sure how a variant that is hospitalising and killing people can be considered coming to our rescue. The daily death rate in Australia is currently the highest it's ever been. And if something was going to come to our rescue, the right time would have been at the start of the pandemic, before over 5 million people died.
Ghost writes, now someone else still has some specimens of the Wuhan strain and may decide to experiment on it to make more transmissive, but at the same time less pathogenic. Viruses less pathogenic. In other words, what Ghost is saying here is saying that someone's probably got hold of the original virus, the B11 from Wuhan. That is possible. And he's saying that just as people did allegedly, or may or may not have done gain of function research, it's a controversial issue, could people have done reduction of function research to actually make a viral saviour? To tell you the truth, I don't think people are that clever, but it's an interesting idea. Then release it in South Africa, knowing it will outcompete Delta. Which animals might have been used in a laboratory? Which animals might have been used in a laboratory setting to make a new variant? Which animals might have been used in a laboratory to make a new variant? I wonder if it could have been mice. Maybe that's what Ghost is thinking, but of course it sounds like he's very cynical.
Okay, as I've previously mentioned, Omicron didn't evolve directly from the Wuhan strain. B11 is not the Wuhan strain. So starting with the Wuhan strain in a lab wouldn't have produced this variant. Now, John does say that he thinks this is unlikely because scientists aren't that clever. I would say it's unlikely because scientists aren't that stupid. Greater transmissibility is no guarantee that one variant will replace another. The variant that wins is the one with the best fitness. This could be the variant with the higher transmissibility, but not necessarily. If we look at influenza, for instance, the new strains that appear each year don't have better transmissibility. They just have immune escape. And there are now 2 pieces of research that suggest the same could be the case with Omicron. In this paper, which was published in Nature, they analysed the data from the beginning of the Omicron outbreak in South Africa. And based on some fancy modelling, which I don't really understand, but I'm sure the reviewers did, they determined that the spread of Omicron could be entirely explained by immune escape without any actual increased transmissibility. Now, this is modelling, so it's not definitive, and the authors acknowledge this, but it is certainly possible. This paper is still a preprint, so it hasn't been peer-reviewed. But in this paper, they compared the rate of secondary attack in households where someone had been infected with Delta to households where someone had been infected with Omicron. And what they found was that in unvaccinated households, there was no difference in the secondary attack rate between Delta households and Omicron households. In contrast, in vaccinated households, the secondary attack rate was higher in Omicron households than in Delta households. So this again suggests that Omicron is spreading as a result of immune escape and not as a result of increased transmissibility. And just in case you're wondering, the study also showed that the secondary attack rate was higher in unvaccinated households compared with vaccinated households and lower in boosted households.
So mice are used in large numbers in lab research in coronaviruses. It's true, they are. Wuhan Institute of Virology used so-called humanized mice, and we did look at this, genes from humans that have been put into mice, and I expressed discomfort with that. Strange correlation, why mice? Next lab leak will destroy humanity. Let's hope not, but these are serious issues. This is why I'm reading them out.
This claim is totally nonsensical. What the person is referring to as humanized mice are mice that have been genetically modified to express human ACE2. As John ably explained in his previous video, the Omicron variant appears to have mutated to bind better to mouse ACE2. So you wouldn't create it by working with mice that are expressing human ACE2. And just on John's discomfort with what he refers to as humanized mice, I just wanted to point out that this isn't something new and it isn't just used in virus research. It is also widely used in cancer research and research into many other diseases. I personally don't do it myself. I just work with cells, but it is necessary. And if it wasn't done, we wouldn't have half of the medical breakthroughs that we have today.
Rich talks about a fourth hypothesis for the origin. If engineering a virus for a gain of function, wouldn't it make sense also to engineer a virus with loss of function that is highly infectious as a safety valve? Interesting. So in a future pandemic, could we have a virus which is circulating, which is transmissible and very pathogenic, then engineer a virus which is less pathogenic but more transmissible that will outcompete it, and instead of vaccinating the planet, expose the whole planet to a virus which is less pathogenic but more transmissible, using one virus to protect us against another virus? Very interesting idea, Rich. Very interesting idea. Ed, fourth hypothesis again, Omicron was created in a lab using lab mice, engineered to give the combination of high transmissibility and low sensitivity. So a lot of people are saying the same thing.
No one would deliberately try to make a virus that was more transmissible but less virulent because it's a stupid idea. There are so many things that could go wrong. Firstly, you would have no way of knowing that it was definitely less virulent without actually testing it in people. And if it turned out not to be less virulent, it would be too late because it would be already spreading. Secondly, even if it started out less virulent, there would be nothing to stop it mutating to a more virulent variant that was still highly transmissible. Thirdly, as we previously discussed, being more transmissible doesn't mean it will necessarily displace the other variant. It's much more complicated than that. Fourthly, if you were trying to make a virus that was more transmissible to humans, you wouldn't try to make it by trying to adapt it to a mouse because you'd expect that to make it less transmissible to humans. Finally, you can't actually do research with animals without getting ethics approval first. So even if one crazy person thought this was a good idea, they wouldn't be able to actually do the research.
Nancy, sounds like more gain of function where they used humanized mice. We don't have the data to answer this.
As we've previously discussed, we do have the data to answer this. It didn't come from humanized mice.
And Dwight says that explains my sudden urge for cheese.
Finally, a worthwhile comment. I love dad jokes, and that was a good one. Now, I just wanted to finish by saying this video is a critique of the information that John has been sharing on his channel, not a critique of John himself. I really like John, and I think he is a good person and a great teacher. Some of you may know that I used to appear on his channel, and he was always delightful in all my communications with him. But he does sometimes share information that isn't true, and that's what this video was designed to address. If you would like to look further into the data that I've presented, I've provided links in the video's description. And please remember, this video is about science, but you shouldn't take it as medical advice. For that, you should speak to your medical practitioner. Thank you for listening. If you found this video useful, please hit the like button so that more people will see it. And if you'd like to see more videos about the science in the future, please hit the subscribe button.